during the because of lack of time, we have to move on, but there is already one question for you in the chat box, so please answer it there. And our next speaker is Paul Zimba. Paul, are you here?
Yes, I am. Thank you.
Great. Can you share your screen and start your talk?
Attempting to right now.
Great.
I want to thank you. That's working, I assume?
Yeah, it is. You can start.
Thank you. I want to thank the Euglena Network for the opportunity to discuss some of the work that my lab has done with regard to anti-cancer activity of Euglenophysin. And this work certainly was benefited by the amazing resource, I think, of Butos, The Biology of Euglena. I really urge everyone to go back and look at that seminal 2-volume set, because there are so many beautiful bits of information that people overlook, I think, in that amazing book. And the take-home message from this is that certainly euglenoids are very unique: cell wall, the method of storage, and probably most importantly, ability to switch on and off by growing autotrophically and heterotrophically. And this presented a unique opportunity when we observed massive fish kills in autotrophic mode for Euglena sanguinii dominance in ponds. We initially found this in one state, North Carolina, and it was followed quickly by 3 other states, and then a series of culture collections that were of Euglena sanguinii from a variety of countries, all yielded positive results for toxin production. We published the Toxin Structure in 2010, and this was followed up by a second paper where we look for the occurrence of toxin production across the entire division. We analyzed 31 strains, and those that are having a solid circle are those that showed positive activity in terms of, positive toxin accumulation. The euglonales are primarily where we find the toxin, not in the other branches of the Euglenophyta. Really, little else can be said about the occurrence. This suggests one of 2 things: either there's considerably more intricate information that needs to be dug out on the genetic organization of the Euglena, particularly when you consider the heterotrophic component is largely unstudied. Of interest in regard to the toxin formation was the 4 to 5 order of magnitude difference in toxin production associated with strains. And Euglena sanguinii was the dominant producer, certainly, but there were a number of other strong candidates for potentially toxin accumulation occurring in nature. When we were doing this isolation, we used cancerous cell lines, and this provided an opportunity to see a U-shaped response curve associated with those cell lines. This is typical of an anti-cancer activity, and cancer basically can be regulated at one of 2 junctures. The Mtor Pathway is where you have a regulation of cell membrane synthesis, and this is a principal area of managing cancer or attempting to. The other would be in angiogenesis, the formation of blood vessels associated with tumor development. This is VEGF development that controls this. And so on separate occasions, we pursued both of these pathways. A student of mine published a paper looking at VEGF regulation, and you can clearly see that there's a strong logarithmic or curvilinear response to toxin dosing, and this is also has the impact of altering the shape of cells as shown in the right-hand panel, where in panel E you begin to get blebbed cells, very few in number relative to the density in the other panels. This very clearly showed that VEGF formation was being regulated, which could be a way to reduce blood vessel formation in tumors. The alternative, the Mtor pathway, was pursued by a graduate student, Cabana, and published. Then this basically showed that the mTOR protein synthesis abundance, protein abundance was affected by Euglenophysin. Particularly, you can see in panels D and G, where there is a significant decrease relative to the vehicle and the control in regard to the mTOR fold change. This can also be seen in the tumor volumes associated with the application of Euglenophysin to these tumors, and there was a significant reduction in tumor volume associated with the treatment. This again is very positive in terms of outcome. We have also looked at Euglenophysin as an adjuvant, basically as a means of increasing bioactivity or activity in general, not so much bioactivity, of compounds to treat cancer. And this basically has resulted in a 3 to 7-fold increase in anti-cancer activity when these are combined. And this is shown on the left-hand panel, comparing the NP1 versus addition or treatment there. So a bottom line in all this is that our funding has been cobbled together from a variety of sources. We have not had success getting National Cancer Institute or the V Foundation interested in a biological solution. They typically have in our reviews have pointed out that there is no certainty of growing Euglena, purifying Euglena, and having toxin of sufficient concentration to do all the testing that we would like. And so this is a problem. We've tried synthesis, and we were unsuccessful in having 2 commercial labs produce more than 1 milligram of mixed purity Euglenophysin. But this is this funding source and direction is a really key item. I think that it's also very intriguing that the adjuvant activity of Euglenophysin is very strong, and it points out to a secondary role that has not been adequately investigated. Lastly, it's clear in Euglena that there are many compounds having biological activity, and I point out a paper in 2016 showing that lymphocyte activation occurs by exposure to peramilon. And so there are a lot of activities that are associated with euglenoids that we're just barely touching the surface of, and this is a very welcome conference towards that end. I appreciate your time. Thank you.
Thank you, Paul. So if there are any questions, please again ask them using the chat.